Hey, welcome back to the channel. Today we're going to be diving into a paper published by Trinity College Dublin focusing on PD-1 gamma delta T cells and how this checkpoint might shape the course of autoimmune disease, specifically multiple sclerosis, using an EAE model. So if you just take a look at over here, you can see that on our citations page on the Assay Genie website, so just assaygenie.com/resources/citations. We've got a link to this paper here. So PD-1 is a key immune checkpoint that prevents autoimmunity by dampening T cell responses. It's famous in cancer immunotherapy, but it turns out to play a very specific role in IL-17 producing gamma delta T cells. And these cells are early contributors to inflammation in diseases like multiple sclerosis. So if you take a look at some of the figures in the paper, just scroll up here a small bit and maybe just move myself around. I'm not cooperating today. I'm going to make myself a little smaller here as well. Push myself over to the side. So let's go up here. So we're at figure number 2 here, and that's figure 3. I think we're at figure 1 here now. Yeah. Okay, so if you look at figure 1 here, and just take a slight zoom out. This is great work by the Kingston Mills group. And since in this figure, actually, they're shown that PD-1 expression is markedly higher on CD27 minus gamma delta T cells, especially in the V gamma 4 subset, in both the lymph nodes and in the CNS during EAE. Notably, figure 1A and B highlight how CD27 gamma delta T cells outpace even CD4 T cells in PD-1 expression early in the disease. So, great work here actually opening the paper. So this tells us that PD-1 isn't just a marker, it's potentially a control switch here for the most pathogenic T cell subsets. If we move on to figure 2 and 3, so good bit to cover here. Very extensive paper here in the European Journal of Immunology by the group. What's going on here is that in figure 2A, the clinical severity of EAE is significantly worse in mice treated with PD-1. They also lose more weight in figure 2B, and in figure 2C to F, there's a surge in IL-17A, CD27, V gamma 4, gamma delta T cells in the lymph nodes. So blocking PD-1 equals more pathogenic IL-17A cells, which equals more disease. Then in figure 3, if we scroll down a small bit here, give me a chance to scroll down. Lots of great flow cytometry work going on here. You'll see that the story continues in the brain of the spinal cord. In figure 3A shows there's a sharp increase in interleukin-17A, interferon gamma, TBX21 mRNA in the spinal cords of the anti-PD-1 treated mice. So this carries on for figure 3B to G, that there's a corresponding rise in these gamma delta T cells, specifically the CD27 minus gamma delta T cells, the CD4 T cells, and a cytokine storm of interleukin-17, TNF, and GM-CSF in both populations. So, there's a lot happening here. So what we can see here, and maybe to understand the mechanism, the authors have isolated V gamma 4 and V gamma 6 gamma delta T cells and stimulated them with cytokines IL-1 beta, IL-23, or TCR. And the key result from figures 5, C, and D, let's scroll down a bit to see these two. Just give the PDF a second to load. Good stuff. There's some beautiful bar charts. So, the key result from figures 5, C, and D is that the PD-1 agonist, PD-L1-FC, significantly suppresses IL-17A when the cells are TCR stimulated, but not when they're activated by cytokines alone. So PD-1 controls adaptive gamma delta T cells, but innate-like ones, those triggered by cytokines, slip through the net. So overall, like, this is an excellent paper. The key study here is a powerful warning that while checkpoint inhibitors like PD-1 are amazing in oncology, they could amplify autoimmune diseases like unleashing gamma delta T cells. Yeah, so phenomenal work here by the team. Uh, if you found this interesting, we do have a lot of publications on the Assay Genie website for some of the reagents that we have and use, or actually specifically been used by this group. In particular, the Mills group used our antibodies to help with their study. And if we just scroll down to the material methods, we can see that two Assay Genie antibodies were used in this paper, which was excellent. So our ORMP1-14 clone for our anti-PD-1 antibody, and our isotype for this clone, the IgG2A isotype control. And you can find these on the Assay Genie website. You can see it here for the PD-1. So here's a PD-1 and the IG2 control, so IMVB0198 for the control, and then IMVB0037 for the antibody. So that's everything for today's video. I think thanks for watching.